This is Dr. Rodriguez, Psych 101, Chapter 9, Development. So this chapter is looking at how we change and how we stay the same from conception until death. It looks at how we change and stay the same in terms of our body, our brain, our senses, our motor skills, health and well-being. It looks at cognitive aspects, including learning and attention and memory and language and thinking and reasoning. So all that cognitive, that thinking part of us. And the psychosocial parts of us refers to our emotions, our personality, and our social relationships. So how do we change and how do we stay the same physically, cognitively, and psychosocially? So how are these topics studied? You can study it through observation, case study. An example might be Jeannie, the little girl who was found after she spent, I think it was 12 years, being essentially locked away with a lack of social interaction. So they studied her, they observed her, they watched her, and over the years to gather information. You can do surveys, you can do experiments, you can, different ways that we study other psychological concepts is how we study lifespan development. All right, so terms that you need to know for when we are measuring development is cross-sectional. Cross-sectional is, say you want to know how people's happiness changes or stays the same throughout their life. In cross-sectional, what you do is you go across the different age sections in society. So you go get some people who are 5 years old. You go get some people who are 10 years old. You go get some people who are 15 years old and some people who are 20 years old. And you measure them. Maybe you give them a psychological test where they self-report their levels of happiness. And then you compare how happy the 5-year-old is compared to, say, the 15-year-old. So a cross-sectional is kind of done at one point in time, and you're comparing the different sections, the different age groups. Now, longitudinal is different. It is following the same subjects over the long haul, over a long period of time to see how they change. So maybe you start off with a group of 5-year-olds and you measure their happiness, and then you come back 5 years later, and you test those same people again when they're 10 years old. And then you come back and you test those same people again when they're 15. And that will let you know how their maybe happiness changes or stay the same. So with longitudinal, I always remember that you're in it for the long haul. You're going to be following these people for a very long time. It's not just done in one day by handing out a psychological test. It is following these people, retesting them after years go by. And then cross-sequential is a combination of both. So maybe you get some 5-year-olds, some 10-year-olds, some 15-year-olds, you give them the psychological test, and then 5 years later you come back, and when the 5-year-olds are 10, you retest them. The 10-year-olds are 15, you retest them. So it's just a combination of the two. So cross-sectional is going out and getting some 5-year-olds, 10-year-olds, 15, getting some different age groups and comparing them. Longitudinal is getting a group, and then you follow them throughout their life to see how they change or stay the same. Okay, so getting right into it, and we're looking at prenatal development. So what happens from conception to birth? So here you have little egg, right? It's inside the mom. The egg is released, and then sperm is introduced. So you have this 23 chromosome egg, this 23 chromosome sperm. These two combine and create a fertilized egg or a zygote. All right, it's a 46 chromosome single cell. It's a fertilized egg. So the different stages of prenatal development. Maybe in everyday, speak, you have heard of trimesters. These stages in developmental psychology are different. It's not trimesters, okay? It's 3 different stages of prenatal development. And it's germinal, embryonic, and fetal. The germinal stage is from conception So when the egg and the sperm combine, to about 2 weeks. And that little egg sperm, the fertilized egg, is called a zygote. And what's happening during this germinal, this first 2 weeks, is that this fertilized egg is moving down the fallopian tube to implant into the uterus. So it's moving on down. It is getting bigger. It's getting more, complex, but it's really just a lump of cells at this time, and it's just moving on down to implant into the uterus. And when it implants into the uterus, it then moves on to the embryonic period. The embryonic is from 2 weeks to about 8 weeks. So when a woman misses her period, and to then about 8 weeks, and it's called an embryo during this 2 to 8 week period. It's an embryo, not a zygote anymore. It's an embryo. And what's happening here is that major organs are forming. This is a real critical period for organ development or structural defects. So if a woman drinks, does drugs, is exposed to some different teratogens are any substance that can create defects in the baby. This is a real critical time for a baby's development, for the organ development, for the embryo's organ development. Then fetal is from about 8 weeks to birth, and it's called a fetus during this time. And this is when the organs continue to grow and become refined and become functional. This is a critical period for more functional defects. So if a mother is exposed to teratogens during this time, the organs may not function correctly. All right, so the way that I remember these stages of prenatal development is grow ever faster. That little lump of cells, that little fertilized egg, it's just growing ever faster. Think about towards the end of the pregnancy, a baby is putting on, they're putting on pounds. They're developing so quickly. The organs are developing. So grow ever faster, and that'll help you to remember. Germinal is that first 2 weeks when a woman doesn't even know she's pregnant yet. She hasn't even missed her period by this time. Now, during this germinal period, 50% of pregnancies end without even realizing it, because a woman hasn't even missed her period yet. Now, during this time when there's cell division and more cells are being created from this fertilized egg, this is when identical twins can happen. So you have this little zygote, this fertilized egg, and it can split in half and then become 2 identical babies. All right, so that's identical twins have identical genetic material. And fraternal twins, what those are dizygotic, means that there were 2 eggs. Di means 2. There were 2 little eggs inside of the mother, and usually only one gets released, but sometimes 2 can get released. So there were 2 little eggs that were released, and 2 sperms fertilized them. It's essentially having siblings inside of a mom at the same time. So monozygotic is an identical twin, mono meaning 1. There was 1 little fertilized egg that split into 2, and dizygotic means that there were 2 eggs, 2 sperms. It's essentially siblings living inside of their mom at the same time. Monozygotic twins or identical twins are always the same sex because they come from the same fertilized egg. So grow ever faster is that germinal, the embryonic, and fetal. Germinal, it's a zygote, it's the first 2 weeks. Embryonic, it's an embryo, it's 2 to 8 weeks. Fetal, it's a fetus from 8 weeks to birth. Germinal, it's moving, the little fertilized egg is moving on down and becoming implanted into the uterus. In the embryonic, you start seeing some formation of the major organs, and then in the fetal, these organs become functional. In development, you often hear about nature and nurture. Is somebody's personality nature, a result of their genes, their biology, or nurture, how they were raised? So the question of nature or nurture, really, it's not a nature or a nurture, it's nature and nurture. Nature, our biology, our genes are influenced by our environment. There's this turning on and this turning off of our genetic material. And remember epigenetics? I had introduced that really quickly a couple chapters back. So nature and nurture. You have your genes, but then you have the environment that kind of turns on and turns off genes. So our environment shapes us as much as our biology does. Okay, prenatal personality development. All right, so when a baby is born, you can see they have a temperament. You might have heard you being described when you were a baby, you were so easygoing, or you were difficult, or you were slow to warm. What that means is that if you were easy, your temperament was very easygoing, easy, you pooped and slept and ate about the same time every day. It was very predictable. You're very happy, very easygoing, adaptable. Maybe could be passed around, from person to person. Could go on outings. It wasn't a big deal. It's a very easy, temperamented baby. Now, difficult is the opposite of that. Very unpredictable feeding schedule, sleeping schedule, eating schedule. They may cry more. They may have a hard time adapting to change. And that's about 10% of babies. The majority of babies are easygoing, about 40%. And then there's some slow to warm, and that's where they may act difficult at first, but once they get to know somebody, or once they are kind of eased into a situation that they're unfamiliar with, then they start to warm up to it. So it just takes a little bit of time. They may seem difficult at first, but with a little bit of time, they seem more easygoing. So that's slow to warm, and then other. So our temperament later develops into more of our personality, a more complex personality. So our temperament is biologically based. It's inborn. We come into this world with a certain temperament, whether it's easy, difficult, or slow to warm. All right, so looking at development in infancy and in childhood, what's going on in terms of physical development? Well, babies are born with reflexes, like the grasping. If you put your finger inside of a baby's hand, they'll often take their fingers and wrap it around your finger. And it might be like, oh, the baby loves me. Oh, look at them grabbing my finger. In reality, it's just a reflex, all right? They just grasp. They grab a hold of. Rooting is a reflex that they're born with, and that's if you rub their cheek, they'll turn to that direction. Essentially, they're looking for a nipple to be able to eat. Startle reflex, when they hear a loud noise, they startle. So babies are born with these different reflexes that over the first year, they lose those reflexes. These are things these innate reflexes, these innate behaviors that they do to help them be able to survive. Now, looking at gross motor development, gross means big. So your big muscles, what's happening is, they're starting off with not being able to do too much in terms of controlling their muscles. Then about 2 months, they can lift their head. 5 to 6 months, they can sit by themselves. 6 to 10 months, they can pull themselves up to a stand. 8 to 9 months, they can walk with holding onto somebody's hand. About 12-ish months, about 1 year old, they take their first steps. About 17-ish months, they can walk. Now, these are all estimates, all right? Some babies will do it earlier. Some babies will do it later. These are just estimates. So there's no, concern if the baby isn't, say, walking at 17 months. It may just, be that they need a couple more months. Fine motor is more of like tiny little muscles, like hand muscles, ability to draw and write. So that develops over the years. By about 4-ish, 5-ish years old, you can start, the little ones will be drawing pictures that you can start to make out what the picture is. In terms of sensory development, touch is most developed. Vision is least developed. They can see best within about 12 inches. And if you think about 12 inches, that's about from the mother's breast when they're feeding the baby to their face. So it's about 12 inches is where they can see best upon birth.